My proposed intervention is to use heart rate variability biofeedback to enhance emotion regulation in younger and older adults. It turns out vagal heart rate variability at rest is one of the best predictors of emotional well-being and emotion regulation ability. You might be asking, what is vagal heart rate variability? It is not just any sort of random variability in heart rate, but it is an oscillation at the frequency of breathing. So this is a recording from someone in our lab who sat quietly for 4 minutes, and we recorded her heart rate. And what you can see is that every time she breathed in, her heart rate went up, and every time she breathed out, it went down. And you can see this very steady oscillation in heart rate. And this is a sign of someone very healthy and also a sign of good emotional well-being, this steady oscillation that relates to breathing. And it's called vagal heart rate variability because it reflects signals that only the vagus nerve can transmit that quickly about the breathing rhythm that's going on. And this is because you usually breathe at a rate around 3 to 4 seconds. Sympathetic influences are slower than that. And vagal heart rate variability is usually measured with high frequency heart rate variability or the root mean square of successive differences. In our research, we have found that vagal heart rate variability, individual differences in vagal heart rate variability, are significantly associated with functional connectivity in the brain between medial prefrontal cortex and the right amygdala. So by functional connectivity, I mean just that these two regions are more correlated in their activity in people with high heart rate variability. Both older and younger people with high heart rate variability show a significant correlation between their brain functional connectivity in this emotion regulation network and their heart rate variability. We've also seen interesting relationships between brain structure and heart rate variability. In both older and younger adults, we see that heart rate variability is associated with greater structural thickness in a part of the prefrontal cortex that is very important for controlling heart rate variability and coordinating the oscillations in one's heart rate. And these findings that we've seen in our lab have since been extended in a very large sample across many labs across all of these participants. In addition, in a meta-analysis of MRI studies, it was shown that vagal heart rate variability is associated with cerebral blood flow in a variety of regions also associated with emotion regulation, such as medial prefrontal cortex, the anterior cingulate cortex, and the amygdala. So what we have across studies is that heart rate variability seems to be a really good metric of how the emotion regulation circuits in the brain are functioning, giving us a useful indicator of the structural integrity and the functional connectivity within key nodes of emotion regulation network. But our question is whether it plays any causal role in enhancing emotion regulation network function. So can we see the pathway from the heart rate variability back to the brain, where heart rate variability is actually influencing brain structure and function? And we do have some hints in the literature that this might be the case, because studies have started looking at this novel treatment called heart rate variability biofeedback. And this is where people learn to increase the oscillations in their heart rate quite dramatically for short sessions every day. And doing this for about 5 weeks tends to reduce depression and anxiety with a large effect size. So it looks like training your heart to show greater oscillations can actually do something to the circuits in the brain that are involved in emotion. But surprisingly, no studies have examined whether there are any long-term effects of heart rate variability biofeedback on the brain. So our hypothesis is that sessions involving high amplitude oscillations in heart rate will strengthen regulatory control over brain activity in emotion-related brain regions. Now, we're also interested in ruling out some possible confounds, because it could be that heart rate variability biofeedback could be due to a placebo effect, or just the time spent relaxing. So in our planned study, we will be examining how increasing the amplitude of heart rate oscillations every day for about 20 minutes could have a positive effect on emotion regulation circuits in the brain. In our main target condition, we will have participants use what is actually a commercially available product called HeartMath M-Wave, where people get biofeedback about their current heart rate. So this is someone's current heart rate, and you can see it is oscillating quite dramatically, somewhere between around 65 beats per minute to around 80 beats per minute. And the reason that it is oscillating so dramatically is that this person is breathing to a metronome that is slowing down their breathing. So they're trying to breathe at around a 10-second rate, which is a slower breathing rate than is typical. I told you a typical rate is 3 to 4 seconds. This person is breathing in every 5 seconds and out every 5 seconds. And they're doing this biofeedback while wearing an ear clip that measures their pulse. And that's how the computer gets their signal as to what their current pulse is. And they're also looking at a metronome that is telling them what pace to breathe at. Now, why does slow breathing lead to such strong oscillations? It turns out that breathing is not the only signal that leads to oscillations in your heart rate. Another really important signal is your blood pressure feedback system, which is known as the baroreflex. And this system has a fixed frequency that is around 10 seconds. And if you slow down your breathing to that same frequency, you end up showing really large oscillations in heart rate, which is what this person is showing here. Much larger than typically at rest when you're just breathing normally, because you have two signals perturbing the heart rate at the same frequency. So as I mentioned, we really want to try to avoid the confounds that are involved with any intervention, where it could just be a placebo effect, or it could just be the effects of relaxing. So we designed a unique comparison group that is trying to do the opposite thing, which is to try to decrease their heart rate variability using biofeedback. And we reasoned that we could tell participants that they would be trying to maximize their calmness to try to get a steady and slow heartbeat. And we've seen in pilot work that participants are just as motivated and involved in the biofeedback in both groups, and also spend as much effort and time doing the biofeedback. We will compute this, again, using biofeedback from the person's ear and the same actual equipment, and we'll give them basically the opposite of the feedback that they get in the other group. So we plan to test 106 younger adults, 106 older adults, and we will randomly assign them to either the increase oscillation or the decrease oscillation condition, with the constraint that 50% of each group will be male, 50% female, and we will be recruiting them to be representative of LA County in terms of race and ethnicity. We have an intensive intervention schedule planned where participants will be enrolled in the study for 7 weeks, and they will do 5 weeks of home biofeedback. The first 2 weeks, they'll do a lot of baseline measures, including MRI, and then the final 2 weeks, they'll repeat those baseline measures, including another round of MRI. And at home, they'll be doing biofeedback every day, and we will set up the study to give them reminders so that we make sure that we have good adherence to our intervention and that they're actually doing it. Now, our primary outcome will be based on our research, finding these individual differences, relationships between heart rate variability and the functional connectivity within emotion regulation network. And here, this is just an individual difference relationship. We are predicting that by manipulating heart rate variability, we can also manipulate the degree of functional connectivity at rest in this circuit. So what our prediction looks like is that the increased oscillation group will show more amygdala prefrontal resting state functional connectivity while at rest after 5 weeks of biofeedback training than they did before, whereas the decreased oscillation group will show no significant change on this measure. We have a secondary outcome too. As I mentioned, we've seen these interesting structural volume correlations between heart rate variability and medial prefrontal cortex, ventromedial prefrontal cortex, or the orbital frontal cortex. So we are predicting that left ventral PFC will show increased structural volume in the increased oscillation condition, but not in the decreased oscillation condition. So this study fits into a broader context where previous studies have shown that heart rate variability biofeedback reduces anxiety and depression, but has not actually demonstrated how this happens and has not even collected any brain measures of what happens to participants when they do heart rate variability biofeedback. Prior studies have also shown that individual differences in heart rate variability are associated with individual differences in brain structural volume and functional connectivity, but we don't know, is heart rate variability just a signal of a healthy brain, or can it actually enhance brain function? So in summary, our question is whether heart rate oscillations during breathing play any causal role in enhancing emotion regulation network function. And we're also very interested in whether there will be age effects here too. We're predicting that older adults will also show significant positive effects of the interventions. And if this is the case, this has really important implications because older adults generally show reduced parasympathetic system integrity, they show reduced heart rate variability, reduced vagus nerve function. And so if we could increase that function and perhaps even tamp down hyperactivity in the sympathetic system, this could be really important for older adults, not only in terms of their emotion regulation, but also their brain health and cardiovascular health. This study has some very specific predicted findings, but even if we don't obtain those predicted findings, we believe the findings will be highly informative because they should help us identify the brain mechanisms involved in the positive effects of heart rate variability biofeedback on emotional well-being.